HeLa refers to the name of a patient, the treatment of a
Cells had the
cervical
extraordinary ability
carcinoma.
to survive in a petri dish.
Those cells
But
came
while
from this
millions
woman,
used her cells and
Henrietta
billions of
Lacks.
dollars changed hands, her family never knew about it.
We owe a lot of gratitude for what Henrietta provided.
Since 1878, when French physiologist Claude Bernard first proposed that the cells of a plant or an animal could be maintained after death, scientists strive to preserve cells in vitro, or outside of the organism. Cell culture, the process by which cells are grown in controlled conditions, was seen as fundamental to developing an understanding of how the cell developed and functioned. Thus, the way was paved for Ross Granville Harrison, widely considered the father of cell culture, to develop a technique for cultivating frog nervous cells in 1907. French scientist Alexis Carrel, working at the John D. Rockefeller Center for Medical Research, believed cell culture would allow him to discover what affected a cell's lifespan. In 1910, he started a strain of chicken heart cells. His alleged immortal chicken heart was ultimately proven fraudulent, but his technique of subculturing, or cutting an original culture into multiple pieces so that it had more physical freedom to grow, did foster the concept of immortal cell culture strains. By the 1940s, scientists were on the verge of unlocking the potential of cell culture. Polio had been widespread in America since 1916, but the number of cases was peaking, and society was calling for a vaccine. Such a vaccine would have to be tested on cells infected with polio, and cell culture offered the chance to grow cells with the virus. Polio wasn't the only disease demanding cells for research, however. There was no treatment for cancer, and at Johns Hopkins University in Baltimore, Maryland, Dr. George Guy was determined to find its cure. Cell cultures had transformed from something to be viewed under a microscope to valuable tools in combating the world's most aggressive diseases. Guy and his wife, Margaret, believed that an immortal human cell line could be manipulated and experimented upon by scientists to find a cure for cancer. He had been collecting samples from patients at the hospital, and in 1952, he began to culture the cells of an African-American woman named Henrietta Lacks. Henrietta Lacks was born on August 1, 1920, to poor tobacco farmers in Roanoke, Virginia, and later moved to Baltimore County, Maryland, where she raised her 5 children. At the age of 29, Henrietta felt a tight knot in her womb. She visited the Johns Hopkins Hospital, where she was diagnosed with cervical cancer. In February of 1951, at the hospital's colored ward, a doctor performed a biopsy, took pieces of her cervix tissue, and gave them to Guy, without Henrietta's permission. When Henrietta's cells arrived at Dr. Guy's lab, his assistant, Mary Kubitschek, was the first person to culture them. She cut the tissue into 1 millimeter squares and dropped them into pipettes labeled HeLa.
So we would get samples of different areas from patients out of the operating room. Dr. Guy would bring them down, and I would cut them up and put them into tissue culture. And there had been a succession of those, and of course the normals never grow until we got samples from Henrietta Lacks. And her tumor cells just started to grow, and they kept on growing. And I didn't realize what a big thing it was. But Dr. Guy was very quietly optimistic.
After several days, there were millions of cells. On October 4th, 1951, Henrietta Lacks died, but her cells, now in the hands of George Guy, kept growing. Excited by the implications of this discovery, Guy sent these cells to laboratories all over the world. Their rapid growth rate and cheap cost made them easy to mass produce. Thus, these remarkably endurant cells would be used in countless studies. In 1952, Jonas Salk successfully used them to test his polio vaccine instead of the monkey cells he had previously been using. HeLa cells would also be amongst the first individual cells to be cloned and used to further studies on cancer, gene mapping, and AIDS. The status of HeLa cells as a turning point in science is indisputable. For the first time, scientists could assess human reactions to different drugs and environments without using actual test subjects.
HeLa cells are very widely used
use in industry, and the reason why is because they're immortal and they're consistent. So in each lab, we all have different protocols that we write for growing cells. Some people may let a cell sit on the bench for 10 hours. Other people may never let it sit on the bench. The advantage with the HeLa cell is that they're consistent. HeLa cells is an industry standard. Every lab should be able to have it. Every lab should be able to know how to grow it and test it.
But to only consider the magnitude of his discovery is to ignore the fact that Henrietta did not give permission to Guy to use her cells, and how, for 2 decades, her family had no knowledge of the cell's prevalence in science. From a modern perspective, this raises the questions of whether it was justifiable for Guy to have developed HeLa cells without consent, and whether it is fair that the family has not shared the profits of what has become a multi-million-dollar industry. However, in 1952, the prevailing attitudes in science did not hold Guy or Hopkins accountable for the ethics of his research. Guy's actions are completely reflective of society's history of leaving poor, uneducated, often minority individuals in the dark about the research being conducted on their bodies.
In this field, you have this notion of vulnerable subjects, people who are, in a sense, who just don't understand what they're getting into because of their poor education, and they don't understand the provisions of consent.
Guy and Hopkins' failure to alert the Wax family about the use of her cells is indicative of the status of vulnerable subjects in research at the time, a status that is most evident in the Tuskegee syphilis experiments from 1932 to 1972. For 40 years, the U.S. Public Health Service, PHS, studied the effects of syphilis on approximately 400 men. Many of the participants were illiterate and most were impoverished black sharecroppers. The PHS withheld treatment from these men to observe the course of the disease. The experiment was terminated when it was publicized as an alarming breach of the subject's rights. As was the case with Henrietta, at the time, there was no established laws protecting the subjects of clinical research. A turning point in ethics would come when, prompted by the fallout from Tuskegee, the U.S. government enacted in 1974 concrete measures to protect research subjects. Yet these regulations continue to be outpaced by the progress of science. In March of 2013, researchers at the European Molecular Biology Laboratory published the genome of the HeLa cell on the internet, with interests of science in mind, but without the consent of the family. Few regulations currently exist governing human genomes, so geneticists are forced to confront a conflict between the advancement of their field and individuals' rights to their genetic data.
We need to make sure that we can exploit scientific progress quickly in order to advance healthcare. Delaying beneficial treatment to people in need would also be an ethical question. I think today we're at a turning point, so we really have an opportunity to develop ethical standards that protect privacy and allow valuable research to benefit people everywhere.
This recent incident expresses at the same time all that is remarkable about HeLa and all that makes Guy's cells so controversial. On one hand, Guy's success in the culture of human cells was a necessary progression and one that has benefited all of mankind. It provided for many of the medicines and vaccines we depend upon today. Yet at the same time, it stripped Henrietta of power over her body, in a breach of morality reflective of the Tuskegee syphilis study. So while a distinct turning point in science, and despite its potential to have a twofold impact, Guy's culture of HeLa cells did not catalyze a turning point in research ethics. Today, however, the return of HeLa to the forefront of medicine is presenting doctors with a second window of opportunity, a second chance to build on Guy's impact on medicine with a new impact on ethics. As science breaks the frontier of genetics, perhaps the backlash to the publication of the HeLa genome will encourage scientists to treat human genomes with increased sensitivity to their owners and policymakers to enforce this sensitivity. And as society pushes into uncharted territory, the Wax family hopes that we bear the story of Henrietta in mind.
HeLa cell is the gift that keeps on giving, and us as a family want our family, the generations after, and therefore to continue her legacy.
This legacy is twofold. The story of HeLa will remind us of the promise of scientific discovery, but the story of Henrietta must remind us that, above all, we must respect fellow humans.